All right. Welcome, everybody, to this special event on the Nobel Prizes of 2024. The 2024 Nobel Prizes, more than any other series in the past, spotlights the reciprocal influence between natural sciences and AI. In other words, how did deep understanding of physics help development of AI algorithms, and how some of the new developments in AI are enhancing breakthroughs in scientific domains, especially in the natural sciences. So that said, these are things never in isolation, right? AI alone does not solve the problem. There is a whole ecosystem that is supporting AI, and a whole ecosystem of scientists, science instruments. So we have two distinguished speakers today and two additional panelists who are going to share their insights about these prizes. And each of these speakers and panelists have done very deep work in their domain. They're experts in computational science, they're experts in the domain, and now they're using AI, right? So that is why we've chosen our distinguished colleagues. So the first talk will be given by Professor Charlie Brooks from the Department of Chemistry. Followed by that, we have Professor Veera Sundararaghavan from the Department of Aerospace Engineering. So then we are going to have a panel with Charlie and Veera to be joined by Professor Indika Rajapakse, who is from the Department of Computational Medicine and Bioinformatics, and Professor James Wells from the Department of Physics. With that, Charlie, if I want to introduce Charlie, it's going to take me about 30 minutes, which is the length of his talk. So I recommend you go to his lab's webpage and see some of the incredible contributions Charlie has done over the past 3 or 4 decades of his leading research career. And in the upcoming MICDE magazine, we will have a feature on Charlie's research career and research profile. So please read that as well. With that short introduction, Charlie, please, floor is yours.
Thank you very much. It's I must say, an intimidating task to talk about Nobel laureates. But I have maybe the slight advantage is that I've known 2 of them throughout most of their careers. And so what I'd like to do is tell you from really the perspective of someone who's seen these folks develop and see their careers develop and see the machinery and the data and the physics develop around them to bring them to the point that they were as productive as they were, and give you maybe a personal accounting of that. This is why I titled the Nobel Prize in Chemistry Standing on Shoulders, and It Takes a Village, and I'll try to provide you with really some input and some insights into what that village was and whose shoulders they're standing on. So let me begin by introducing, as you know, David Baker was the half winner, i.e. half the prize for his work on the use of computation methods for protein design. Demis Hassabis and John Jumper shared the other half for their work on bringing AlphaFold and machine learning to protein structure prediction to the point where it's now very, very robust as a tool for predicting protein structure. Who are these folks? David Baker has been a Seattleite basically most of his life. He was born in Seattle. He went to undergraduate at Harvard and majored in biology and graduated in 1984, as it's noted. He got his PhD from Berkeley in biochemistry, studying protein transfer and trafficking with Randy Schekman. And this gave him really, as I think was evident throughout his career, a very strong grounding in understanding how biology and structure interacted with one another and how you could use structure to control biology. After his PhD, he did postdoctoral studies at the University of California, San Francisco with David Agard, a structural biologist there, and then moved on to a faculty position at the University of Washington, where he spent really all of his career. He was brought into the Howard Hughes Medical Institute as an investigator in 2000. He was elected as a member of the National Academy in 2006. He was elected as a member of the American Institute of Arts and Sciences in 2009. I will point out, and I'll bring this back up a little bit later, that his first protein structure prediction paper occurred in 1997, so really about 4 years or so after he began his career as an assistant professor. I met him shortly after that, actually, at his first participation in CASP in 1998, and CASP is the critical assessment of structure prediction, where really a lot of the ferment and growth of methods for structure prediction occurred over the last several decades. Demis Hassabis was born in North London and grew up there. He got a master's degree at Queen's College in Cambridge in computer science. He went on to do his PhD in 2009 from University College London, UCL, where he studied both neuroscience and aspects of AI. He served a stint as a visiting scientist at MIT and Harvard. He went back and did some postdoctoral studies at University College London with Peter Dan. Then he moved off to DeepMind, where he is now currently the CEO, and he was the co-founder of DeepMind. He was elected as a fellow of the Royal Society. I couldn't find the date for that. I don't know why. He was knighted in 2024. His first efforts in CASP and the birth of AlphaFold were in 2016. The year that CASP and AlphaFold, or the year that AlphaFold 2 made all of its splash in protein structure prediction, and really the sort of revolution to where we're at right now was in 2020. John Jumper, the youngest of these, was born in 1985 in Little Rock, Arkansas. He did undergraduate studies at Vanderbilt and majored in physics and math. He did a master's degree at Cambridge in theoretical physics, and then moved on and did another master's degree at the University of Chicago in theoretical chemistry, where he hooked up, I think, with Tobin Sosnick and Carl Fried, and stayed on for his PhD with the two of them. Now, Tobin was a member of a group called the Protein Folding Consortium, which was funded by NSF for about 10 years, that brought faculty together. I was one of the lead scientists on that effort, with graduate students and bringing theorists and experimentalists together to really talk about the methods of protein folding. That is, the study of the folding process, not necessarily structure prediction. I met on a couple of occasions John because he attended those meetings, which took place every year. Anyways, John finished his PhD in 2017 and moved right to DeepMind, where he joined the team working on AlphaFold. And it's his efforts, I think, largely in with AlphaFold 2 in CASP 14, which took place in 2020, which recognized the power of machine learning and the data access that they had available to train those models with winning CASP 2 in 2020. He was recently elected as a fellow of the Royal Society. All right, now, sorry for those of you who don't want to learn anything about biology, or maybe even a little bit about physics, ultimately. I thought I really couldn't do this without saying a little bit about what the ideas behind protein folding are. So protein sequence determines fold. That's really the protein structure prediction sort of question. And proteins are largely organized in a hierarchical fashion, starting with their primary sequence, that is the amino acids. And this just illustrates the sequence of insulin, the two chains of insulin that make up the insulin molecule. It turns out that one of the shoulders on which they stood was the Chemistry Nobel Prize in 1972, which was given to Chris Anfinson for his thermodynamic hypothesis that basically said protein sequence determines structure. So in fact, without that being a truth, of course, trying to fold proteins to the structure from their sequence would not be possible. And of course, he didn't make the thermodynamic hypothesis true, but he won this Nobel Prize for his observations on demonstrating, in fact, that protein sequences were really the only information that was required for proteins to fold. In this hierarchy of structure, the sort of second level is what we call secondary structure, and that's a local organization and involves oftentimes local interactions. And it's comprised of bits that we call alpha helix, beta sheet, and reverse turn. It's relevant that these interactions are all really quite local, because very early on, in the 70s with Chow and Fassman, and later on with Terry Sanowski and Sander and Ross, machine learning methods, very primitive machine learning methods, were being used, in fact, to try to understand how the sequence is related to the local secondary structural motifs that one finds in proteins. So already people were thinking about predicting at least these local pieces of structure using machine learning sorts of methods. And that sort of carried forward as things went on. Tertiary structure is the next level, and this is the sort of overall folded architecture of a protein, and this is a cartoon of a protein in which we see several of these beta sheets, the single alpha helix, and then these turns that join the alpha helix together. And the key thing, aside from really the beauty of these architectures, and I should point out here that one of the things that David Baker was recognized for was the design of a really brand new, not seen before protein fold. It was called Top 7. Early in the process, the fact is it looked much like this, except that it had 5 of these sheets as opposed to 4, but it didn't look like anything else, any of the other structures that were known. But a key component that one takes away from these is how, and we don't see the side chains, the little amino acid side chains that hang off here, how these side chains interact with one another through the structure. So distant in sequence, so this a side chain here, say, interacting with this strand of beta sheet, far away in sequence, but very close spatially together, such that they form some sort of interaction. And it's those interactions which we capture typically by this pattern of interresidue contacts. And here's a contact map. And as I'll point out a little bit later, contact prediction, that is the ability to learn from the sequences where these contacts are likely to occur within a protein, has been very key to the protein, solving the protein folding problem and being able to, in fact, turn sequence information into predicted structures. On up in the hierarchy are what's called the quaternary structure of proteins. And well, this is not necessarily relevant to this, this lecture today. I did want to just be complete and say this is sort of the organization of these protein folded structures into larger functional organizations. And oftentimes these things, this being a trimer, that is 3 repeats of the same protein, 4 repeats of the same protein, this actually is hemoglobin, and 5 repeats of the same protein, are often the biologically functional units. So proteins often assemble into these sorts of architectures to do the work that they need to do in biology. I will say that the sort of latest kinds of models, AlphaFold 3, works to try to predict these quaternary sorts of structures, and how well is certainly it's an important tool and people are using it. Now I want to talk about the other half of the protein folding problem. That is the one that's probably more related to folding diseases, and that is how proteins fold. So the protein topology, that is that structure that I showed you in the cartoon a moment ago for that protein, largely has been shown to determine how the protein folds, the steps it takes as it goes to its native state. So protein folding mechanism, that's what that's called, and errant folding or misfolding actually determine a lot of protein folding diseases, Alzheimer's and many of the diseases that lead to amyloidosis and other sorts of fibril formations. So some of the organizing principles emerged from physics very early on. Leventhal, for whom, in that long list that wasn't going to be shared, I'm named the Cyrus Leventhal University professor. Leventhal in 1969 defined this computational problem really as a challenge to the community, saying that if in fact one had to search through all of these phi-psi angles. These are the so-called the backbone angles of a polypeptide chain. And each angle had 3 distinct states. Then to solve the structure of a 100 amino acid protein would require searching 3 to the 200th different structures. And this led to these ideas of something which is pictured here called a golf course landscape, that you'd just be wandering around in this space, searching for that one set of phi-psi angles that determined the protein fold. Now, Leventhal said that this would take an enormously long time, and of course proteins fold in a finite time. And so this was the conundrum that I think drove the field early on. Turns out that Cyrus Leventhal, and the reason I was able to adopt his name in this named chair that I have, is that he started his career here in physics at the University of Michigan. He then moved to MIT, where he really ran a very large visualization lab for visualization of biological structures. And then following that, he moved to Columbia, where he founded the Department of Biological Science. He was the chair of that department until his passing. All right. Physics also emerged with another sort of unifying principle for folding that helped reconcile why this apparently complex process, search process, occurred very rapidly. And this is called the funneled energy landscape theory or hypothesis that came about in the 1990s. So this really redefined the computational problem of protein folding, positing that this energy landscape was funneled like this, and you went from unfolded or disordered states along a relatively smooth energy funnel to the final folded state, which was the thermodynamic minimum of this free energy landscape. And that is an explanation about why proteins fold quickly. The roughness on this landscape is why, and I'll show you this in the next slide, why some proteins get stuck and go off to form disease states. And some of the key contributors here were Peter Wolinus and Zan Luthi-Schulten and Jose Anushik, who, by the way, was the graduate student of our physics Nobel Prize, one of our physics Nobel Prize winners a minute ago, that we'll hear about in a few minutes. Ken Dill, I was involved in a lot of this early on, Dave Thermali, and Eugene Shuknovich. All right, so this again idea that this landscape controls how proteins fold, and the fact that we have proteinopathies, that is misfolding and errant protein folding, led to the idea, mostly pushed by Chris Dobson and explored with a lot of NMR experiments that he did in the early 2000s, that these landscapes, in fact, were more complex than just this simple funnel landscapes, that there could be rough areas on the way to the folded state that would trap these structures in states, in conformations or states, that would give rise to opportunistic combining of multiple chains and the formation of aggregates, which then went on to form fibrils or amyloid-type structures. I will point out that University of Michigan has an active seminar program and research thrust in many areas of proteinopathies, which in fact are quite interesting and connect to this sort of question. Now, I call this a technical interlude because I want to try to connect this idea of how the sequences connect to these contact maps and what the mathematical or physical framework that was developed to describe that. I want to show you a little bit in the context of the machine learning architectures, which I'm not going to try to explain because I don't honestly fully understand, but I'll show you the pictures that are sort of used today in Rosetta Fold, which is actually the machine learning folding architecture that came out of David Baker's group, and AlphaFold. So let me start with this physics-based sort of Potts models and direct coupling analysis. And this is where sequences and multiple of these protein amino acid sequences for proteins that are in similar families come into play, because it was recognized quite early on, and I'll say a little bit more about that in a few minutes, that given a family of protein sequences that were aligned to best align all of the amino acids that were the same, all of these letters that were the same, one could build a model that looked like a spin model, but a spin in which you now had 21 states, the 20 amino acids, and then something for a gap. And you could learn what this energy function was. And from that, you could make predictions about, number one, how changes in amino acids would change the stability of the protein. But number two, and probably more importantly, by looking at these evolutionary, and it's evolutionary because this is a family of protein sequences, looking for evolutionary correlations between if an amino acid changes here and one changes here, how frequently do they do that? How often are there covariational changes in amino acids? Those are probably indicative of the fact that if I'm standing here, with my one amino acid interacting with another, and I'm going to change this, in order for the structure to stay together, in order to maintain the stability, I better change this one too. So there's a higher probability that if you have two amino acids that are distant in the sequence that are changing, that there's likely an interaction between those two. And that's really the basis of the ability to predict these contact maps in proteins. This again came directly out of Potts models in physics, combined with learning methods and Bayesian approaches for solving this problem. This work that I'm highlighting here actually is work of Terry Hua, and I've forgotten his last name, or first name rather, and Jose Anushik from UCSD in about 2011. And again, I would like you to remember that time scale, 2011 maybe, to come back to, okay? All right, this is a Rosetta architecture. I don't want to say a lot about this other than you see sort of up here a sort of sequence-based things, the MSA, multiple sequence alignment. And a lot of this is operations on sequences and using different components of machine learning architectures to extract information. This is more based on local structure used to predict things like these contact maps, which tell us about how the protein ultimately folds up. And then these are combined with final layers to sort of ultimately yield all atom models. And again, Rosetta and AlphaFold do very similar sorts of things, although the architecture is different. And again, my main point is that we've brought together a lot of information of sequence and structure to bear on machine learning to find the rules or find the outcomes that yield finally three-dimensional structures. And this is actually the structure of AlphaFold 3 with the sort of diffusion methods at the end. Okay. All right, so standing on shoulders, I realize there's a lot of text here, but there's some points I would like to make. The critical assessment of protein structure prediction began in 1994. This was this exercise where they brought people trying to learn this, trying to solve this problem from sequence to structure together twice a year. And they asked them to share their methods and share their results, and they gave them sequences for which they didn't know the structure or the structure was not solved, and asked them to solve those structures, and then those structures were assessed at the meeting. And then the participants got to present how they did it and what happened. Other key organizers in this first meeting were Krzysztof Fidelis, Michael James, Shoshana Wodak, who actually was a student of Cyrus Leventhal at Columbia, and Fred Cohen. Early participants were Martin Karplus, who was a 2013 Nobel Prize winner in chemistry. And the general idea of CASP, I said, was to review the progress and share the algorithms. Now, I'll tell you, well, I'll get down here and I'll tell you. So early CASP meetings focused on predicting structures based on homology, i.e., can we find another structure that's already known that has a similar sequence? And if so, can we take that structure as our template and just replace the amino acids, the side chains that are different? And then it evolved into more complex, what's called ab initio folding, is that just given a sequence, can I fold the structure? the ideas of mapping these co-evolutionary sequence mutations, so covariation in the sequence, happened in the very earliest CASP. Actually, one of the abstracts in the 1994 CASP was about using a neural network approaches to try to do this. We'll see why those largely failed in a moment. I'll try to present what happened that made those things work finally. Yang Zhang, who was a faculty here at University of Michigan for a number of years, was a developer of iTaser, which was in fact one of the most successful protein folding algorithms for a number of years and competed in CASP quite frequently, and quite frequently was in the very top. As I said, Baker first participated in 1998, and I have a little personal recollection of that meeting. So I told you that the purpose here was to share ideas in what the algorithms were that one was using. And David and his group came to this meeting, and they did reasonably well. They didn't win by any means, but when asked about how did you decide which of the many folds, because you particularly make many predictions for a given sequence, you present it as your top model. And it led to a lot of angst and frustration in the field at the time, because his answer was, I have a group of graduate students and postdocs who are very smart. We all sit around and we look at it and we judge the best one from consensus. Who thinks it's best? And they did that with their chemical intuition and their biological intuition and their understanding of structure sort of function relationship. But it was frustrating to the rest of the CASP developers who were looking for algorithmic approaches to do this. So that was my first encounter with David in the context of CASP. Of course, he did make it algorithmic, and actually the CASP people that ran CASP decided that there really had to be an automated server prediction sort of mode so that there couldn't be human intervention to bypass, not directly as a result of David's, but just because they felt that the field should evolve so that the rules were all sort of built in. DeepMind participated in CASP in 2016. AlphaFold emerged as a winner in 2020. That was CASP 14. And the most recent winner, CASP 16, was actually one of David Baker's former students with an algorithm and method called TR Rosetta. So AlphaFold has not won all of the CASPs since 2020. It won that one. But what has happened, and what has happened throughout CASP's history, is that after these meetings, everybody adopts the things which work best in the field. And that's the way science should progress, and that's the way it has progressed, I think, in this case. All right, so what sort of led us to be able to do some of the things that we're doing? The Human Genome Project and advances in DNA sequence technology initiatives vastly increased the number of sequences of proteins that existed. Human Genome Project started in the 1990s. UniProt, which is the sort of library of all of the known protein sequences, was founded in 2002. This is sort of the list in millions of protein sequences that were available. As I said, the first attempts to use sequence evolutionary covariation to predict these protein-protein contacts was in 1993. Way over here, where I don't really know how many protein sequences we had at that time, but it was just a cottage industry for collecting proteins from similar families. Jeff Skolnick, who was Yang Zhang's postdoctoral mentor, a former colleague of mine in the 2000s, used covariation for structure in his structure prediction algorithms. So this idea of covariation and predicting contacts sort of existed for quite some time. So as I mentioned already, Hua, Waite, and Anushik applied sequence covariation to multiple sequence alignments in 2011. So we're getting right about here where we start to have maybe enough sequences to do things using these POTS-like Bayesian and related so-called direct coupling analysis methods. What happened equally, or at more or less at the same time, is the NIH recognized that they wanted to understand fold space, that is, all of the possible organizations of protein architectures to make protein folds. And they put a lot of money into what was called the Protein Structure Initiative. And that really increased the number of protein structures. This started in 1999, and it went on through about approximately 2015. And you see the growth of known protein structures, of the protein structures deposited in the protein data bank, the repository for all protein structures. So the first CASP was held here, and the number of structures that were available were in the hundreds at the time, okay? There was not a whole lot of examples to learn from. Baker and his group started participating in CASP here in 1998. There are more structures and more sequences, but not a lot more. Here's where DeepMind came into CASP. Huge, huge growth in the number of structural data that one had to analyze, as well as the number of sequences. And here's where they win CASP, and AlphaFold 2 wins CASP. So the point I tried to impress upon you in the last two slides is that there was a lot of physics and physical thinking that really drove many of the developments that went into the machine architecture that is now AlphaFold or that is now RosettaFold. But much of that occurred largely in the absence of sufficient data to train this sort of machinery. And it wasn't until very recently that the data was made available through federal funding initiatives such as the Structure Initiative and the Sequence Initiatives. All right, so it takes a village. I've talked about a number of these people as being people contributing at various points in time to advancing ideas that ultimately led to the kinds of machinery that are built into RosettaFold and AlphaFold. Here at the bottom are our three Nobel laureates. And they really benefited significantly from the advances that were made by all of these individuals throughout the time, both the physical theory advances as well as the data and the growth of data advances. So just to remind you, what I've tried to tell you is that in the early 60s and 70s, Levenfeld's paradox provided the challenge for solving protein folding, both sequence and structure, as well as a folding mechanism. Anfinson's thermodynamic hypothesis set the stage to make us believe that there was a sequence to structure relationship that we could solve. In the 1990s and 2000, federal funding agencies were really ramping up the discovery of new structures and the discovery of new sequences. The CASP exercises greatly facilitated the knowledge and the methodology for finding new structures and solving new structures and really building the base for allowing these machine learning approaches to know what to grab hold of. We need the contacts. We need the local structure. We need information about that. And then we need the data to sort of infer that information from. In the 2010s, this contact determination became practical with sufficient sequences through the production of rich multiple sequence alignments. DeepMind moves into the foray in the late 2010s, and they move in, and as we know, they won CASP in 2020, and many other prediction frameworks then evolved from that. Now, with that, these are the people who won the Nobel Prize. I will say that David Baker has, in my experience, a keen sense of how biology works, and the ability then to know what sorts of structures to try to design. And he's done, I think, a great job of that, and really is very deserving. Demis Hassabis and John Jumper they were there and brought the machinery together at the right time for AlphaFold. So, the last thing I want to say as I'm thanking my group is that, I know that many of us are in, a very mixed feeling about the role of machine learning, et cetera. And I've taken to sharing some of my poetry at the end of my talks. This actually is a poem that I wrote, not necessarily about this, but I thought it was quite appropriate. I wrote this when I was feeling sad because my adult daughter had just moved out of our household after living with us for many years, and I was, felt I was getting old and I was lonely. So, but it sort of fits, I think, the way that sometimes I feel about what machine learning may be doing to my field and what some of us might also think about machine learning. It's called Feeling Finite. Once invincible, the principle of continuity, flowing to a beat, life so sweet. Seldom see the barriers in high dimensions askew. There's always a path to somewhere new. The infinite was a dash, to crash or pass without a thought, to the end of the road, to look around the corner. Now I'm a foreigner in this pace, seeking a comforting place. The path is shorter than it ought be. For me, feeling finite in dimensions too big, with too many edges, too many wedges of loneliness, oneness, missing others, among the count of a natural order, and I'm feeling finite near the end. Thank you.
Thank you for the wonderful talk. Veera, as you get set up, I want to ask a couple of quick questions to Charlie, but you should get set up. One is, for a high-level question, because not everyone has seen protein folding, if you can maybe spend a minute or two expanding on the significance of why this is an important problem.
Well, so as I tried to emphasize, but probably not well, that most of our understanding of how proteins work arise from some knowledge of their structure, so structure-function relationship. So the ability to basically design a new sequence and ask what its structure is likely to be, opens huge roads to design problems. And as David Baker won the Nobel Prize for his acuity and acumen in sort of doing and posing those sorts of questions. But again, because so much of the function of biological systems depends upon the structure of the entities, in this case, proteins, the ability to get protein structure is a huge, huge boost across the range of fields, from drug discovery to new medicines to understanding how proteins interact in biological systems, et cetera. I hope that's
a thank you very much.
High enough level answer.
Yeah, so I have more questions, and I'm sure the audience has, but then we'll take those after Veera's talk. Thank you very much, Charlie. All right, our next speaker is Veera Sundararaghavan from Aerospace Engineering. So he works, he's a computational scientist who works on many things, primarily around the application of materials, microstructure, atomistic level calculations, but also quantum computing and machine learning. So Veera is going to talk about how the physics prize was conceived and some of the people and the ideas behind it. Yeah. Thank you, Veera.
Thank you. So let's have a deep dive into Nobel Prize in Physics. One of the reasons I was called for this talk was my article and the conversation. So my talk is basically around that article, what I wrote about the basics of what led to this Nobel Prize. But most of the work by Hopfield and Hinton, who got the Nobel Prize, was done in the late 70s, early 80s, specifically Hopfield networks and Hinton's Boltzmann machines. So I'm going to talk about some basics of those particular networks. I want to give a background on, let me see, so there's one more laser pointer, some background on what Hopfield did. Hopfield was actually a solid-state physicist. His PhD was in solid-state physics on excitons and crystals with Albert Overhauser at Cornell. Same as me, I also did my PhD at Cornell. But then he actually shifted to biology in late 70s. He did a lot of work on hemoglobin and then later went on to do a lot of work on how the brain works. So he's actually a professor at Princeton. Most of his work for the Nobel Prize was done at Caltech. He's a professor in molecular biology, which is very interesting that he got the Nobel Prize in physics. But all his previous papers, one of the most cited papers, are in physics and solid-state physics. Geoffrey Hinton is known by everyone working in neural networks. He's actually known as the godfather of AI. a little bit of a notorious name as a godfather of AI. Lately, he's been very vocal on what AI in the future will look like. But he was born in London, and he moved to Canada. He's a professor emeritus of University of Toronto. He's all-out computer scientist. His PhD was in artificial intelligence in the University of Edinburgh, and then he is a professor of computer science. He did a lot of work with Google. Recently resigned from Google, and he is currently professor emeritus at University of Toronto. So the prize was shared 50-50 in this case. Hopfield got half of the prize. Hinton got half of the prize. The citation reads, the Nobel Prize in Physics was awarded for foundational discoveries and inventions that enable machine learning with artificial neural networks. A very specific terminology. You would realize there is very little physics in this statement. So what I want to do in this talk is link their work to physics, because this sounds like a computer science award. So let's look at, as a background overview, it goes with my article in the conversation. I'm going to talk mainly about some background in physics. I will start with the neuron, and then I'll talk about Ising models, or Ising models as they should be called, and then Boltzmann distribution. All three concepts, Charlie was actually talking about Boltzmann distribution on one of his slides. I'll go through in a little bit deeper on what Boltzmann distribution does. And then I will go on to Hopfield and Hinton's breakthroughs, which are Hopfield networks, Boltzmann machines. I'll talk about backpropagation, and finally impacts and application. One of the quotes of Hopfield I want to bring up is a question that we still haven't answered. How mind emerges from brain is, to me, the deepest question posed by our humanity. How does the mind emerge? How does the thinking, computational thinking that actually emerges from the brain is still an unsolved problem. So this is something that Hopfield tried to address, and that's why he was a professor of molecular biology. You can read some of Hopfield's papers that he actually said he goes to a conference, he gave a talk, and nobody understood what he was talking about in the biology conference. But they were all excited about what he was doing. So we'll talk about first the concept of neural networks, because that is all biology. So in this case, if you think of a neuron, a single neuron takes some input chemical signals. I call it X1, X2, X3, etc. And then it transfers. What you get is an output. Output is, again, a set of signals, Y1, Y2, Y3. In neurons, the input-output is modulated by chemistry. There are chemical signals, the thresholds, the calcium channels, etc. So in this case, because it's such a complex system, it's been simplified to what we call a single neuron model. And many of you working on neural networks think of it as a basic unit for machine learning. We have inputs. Every neuron takes as inputs some x1, x2 up to xn. And then there is a transfer function which basically sums up all the signals with some weights. There is an activation function that thresholds the output, and then you have an activation. So that's a single neuron, but I also talked about some basic background of what led to these discoveries. Was the first perceptron by Rosenblatt was a simple neural network for binary classification. And then there was a paper published called, there was a book, Perceptrons, which led to the AI winter, which is called the first AI winter, which basically blasted this approach. And nobody's worked on this for a while. And then John Hopfield is actually credited with researching this whole field through this Hopfield networks, which I'm going to talk about. He revived the interest in neural networks. In fact, John Hopfield's approach led to this new approach called Boltzmann machines, which turned out to be a generative network. John Hopfield was purely a trained network by Geoffrey Hinton, which is called Boltzmann machines. So that's really the progression, the main progression in this field. I want to talk about the Ising model. The Ising model is based on, it's a foundational physics model. It was invented in the 1920s, and it's based on a spin model of magnetism. So the idea is you have a lattice, a two-dimensional lattice, that has an upspin and a downspin. And within this architecture, you have each spin interacting to its neighbors, but also each spin interacts with the field. So in reality, this model has some additional connections to the field variable. But then Hopfield actually visualized this model as spins being neurons, and the weights between those spins being interaction, the synaptic weights. So there is some similarity between the Ising model of the 1920s in solid-state physics to actually the neural network that was later discovered. One of the things he talks about in his paper is my knowledge of the spin glass lore and a little neurobiology led to the first paper in which I used the word neuron. It's the most cited paper I have ever written. The first most cited paper of John Hopfield actually used this Ising model, which is the Hopfield network. I want to also talk about statistical mechanics a little bit. It's very important as a background. In statistical mechanics, you have the Ising model, which is basically this lattice. You have two spins. Just as a take-home message of what this is. Every spin thinks of how to set its up spin or down spin. Every spin has every site in this lattice thinks of how to set this up spin or down spin, and it makes a decision based on two items. One is it wants to align with your applied field. This is the applied field. So effectively, you're saying this spin has to be up. That's what the applied field is saying. But then its neighbors are saying it has to be down. So this spin is in a conundrum. What should I set? Should I be up as in the applied field, or should I be down like my neighbors say? And that's basically the Ising model. The Ising model sets a system energy that is trying to align with, because there's a negative sign, tries to align. If H is +1, it wants to be +1 because you want to minimize this energy. And if H is -1, it also wants to be -1. That makes this minimum. But it also wants to align with its neighbors. If the neighbor is +1, it wants to be +1. If the neighbor is -1, it also wants to be -1. And what's the solution of this? The solution is that, that's what the field of statistical mechanics says. The solution depends on the global properties of the system, not just the single properties of your single spin, the property of the larger system and how individual objects in that system collectively minimize the energy. So all these spins have to collectively work together to minimize the energy. This is something natural in biology and also in solid-state physics. The systems tend to minimize the energy that by a collective decision. I want to show you some spin models in different fields just to give a motivation. If you're in computer science, you've seen graph cuts. So think of the same Ising model. The top part, the blue part, is your external field. The external field is the actual image. So you want to be as close as possible to the image. So the actual image ends up being the external field. The spin interactions wants to make the image very smooth. So it tries to make sure the next pixel is same as the pixel next to it. So it tries to form a smooth image. Basically a denoising problem. Ising model does a denoising problem. It takes a noisy image. The output will become a smooth image just because your spins want to be same as the pixels want to be same as the pixel next to each other, as well as trying to make sure it matches what your input image is. Another example is, very common example is ferromagnetism. This one is T equal to 0 Kelvin. You want to minimize the energies. Your spins, if you initialize the spins, they try to minimize energy. They all try to line up. But there are antiferromagnets like this where the field perturbations can cause phase transitions. Finally, you have the Hopfield network. In the early 80s, Hopfield came up with this network. He called it associative memory in a sense that the critical phases that form are stored as solutions of this network. So whatever pattern you give, it tries to fit it to one of the solutions, the minima of the Ising model. This is a picture that tells us how the Hopfield network works. The basic idea is we store memories really fast. So if we hear a song, if I hear a song, I know what the song is almost immediately. I know the lyrics to the song. Just from the first few sound bites, I can find out what's the lyrics to the song, what song the radio is playing, etc. This is called associative memory. We associate those musical notes to certain lyrics or certain songs. Because our brain can instantly recall, it knows minima. Because we have these networks, the networks know the minimum solutions. And as soon as we hear a stimuli, the neurons fire in a sense that it gets to the minimum. That's what Hopfield networks does. Hopfield network tries to find certain minima to a system that codes in your inputs. So Hopfield network can only predict what's in the training set. If you give a training set a bunch of patterns, it fits those patterns to a certain minima in the Ising model. It can only predict those patterns. This is called Hebbian learning. It's not backpropagation really, but it's really a special type of learning called Hebbian learning where you take an outer product of your inputs and that generates a certain energy surface. This energy surface of the Ising model is coded in the weights. You change the weights so you actually recall those specific patterns you train. That's really the Hopfield network. I want to jump into what Hinton did. Hinton developed what is called a Boltzmann distribution. So in the Boltzmann, developed the Boltzmann machines. He didn't develop Boltzmann distribution, of course. He took the idea of Boltzmann distribution and put it in a real network. He built onto the Hopfield network in a certain way. I'm going to show you how he did it in simple ways. Again, using the Ising model. You take this Ising model, and then you can say the energy of any state in an Ising model is a Boltzmann distribution. This is a Boltzmann distribution in physics. It's basically an exponential distribution with an inverse temperature. In effect, Boltzmann defined what temperature is. So it's really a pre-multiplier to the energy. And then he defined what temperature is based on that pre-multiplier using what is called a Boltzmann constant. It's an inverse temperature. So what this means is at higher temperatures, as temperature increases, all possible combinations can exist, a noisy mode. If the temperature drops, there are only certain combinations that can exist because the probability goes closer to 1. So there's only 1 or 2 phases that can exist at lower temperatures. In this case, I show you some examples of what Boltzmann distributions have in different fields. So here, a solid state, at low temperatures, if you raise the temperature, it becomes, there's a phase transition. Solid becomes liquid. You see a more energetic system. It's having a lower, all your configurations have higher probabilities, so you have a more complex system. If you sample, you get lots of states. In this case, example in solid state physics, you're looking for minimum energy crystal structures. Some of the things I work on is you're looking for certain crystal structures that have minimum energy. Again, what Charlie talked about is configuration of proteins also have similar features where you look for minimum energy folded protein structures. Finally, I want to jump to restricted Boltzmann machine, an example here, which was Hinton's work that led to a Nobel Prize in physics. What he did was a generative network. So that's a really cool aspect of the extension to Hopfield is Boltzmann machine is truly a generative network, which is very close to what the modern generative networks do. They create new outputs to unseen stimuli. So what he did was he took the Hopfield network, and then he tried to apply the physics of Boltzmann distribution in a sense that he said we can actually learn the probability distribution of data. If you're given some data, it doesn't need to lie in certain minima, but it can follow a probability distribution. With that, if you give new stimuli, you can sample that probability distribution and get new outputs. So it's truly a generative model. I want to also show you a little bit of what this Boltzmann machine does. So you have a network, undirected graph, in which you have some nodes, and there are some visible nodes, which are these 3. 2 of the visible nodes take some inputs. The blue node gets some output. There are 2 gray nodes, which he calls hidden nodes. So hidden nodes are not seen. They don't have any input or output, but they're part of the training process. They're there. They are involved in this joint probability distribution. So there are 2 aspects. One is classification, that you given an input, there is an output that tells you whether it's 0 or 1. It's a classification problem. Or you could do a generative problem. So I want to show you the example of a generative problem is you can create new phases. If you sample this Boltzmann distribution, it can generate some new phases based on some probability distribution at different temperatures. So effectively, what he did was something called contrastive Hebbian learning, which is very similar to the Hopfield network. The learning process is really not back propagation. Back propagation was later popularized by Hinton. But the contrastive Hebbian learning does very cool thing in physics is that when you actually minimize probabilities, you would both minimize the energy of the system, which is what Hopfield did. Hopfield was purely minimizing energy of the system. That's what Ising model does. But with probabilities, you also have to minimize the log of the partition function. So physically, the way you think about this is out of all the combinations of outputs from your Boltzmann machine, you want to maximize the probability of your correct output, but you also want to maximize the energies of all other outputs. When you maximize the energies of all other outputs, you're minimizing the probabilities of all other outputs. So it's like a joint learning where you have to do it in 2 steps. That's why it's called contrastive learning, where you would minimize the energy of the system, maximize the energies of everything else coming out of the system. So I won't go deeper into this, but just want to show you some examples of what can be done with a Boltzmann machine. So it's a generative model. If a part of a picture is missing, so you have a picture, how part of a picture is missing, you can pass it through the Boltzmann machine. Here I call it deep Boltzmann machine. And then it'll output this entire, it'll try to build a generative case, it'll try to build that image back. The analogy to physics is we're always looking for the minimum energy solutions, but in this case, we are similar to crystal structure prediction, we are looking for some minimum energy solution for this problem. It's looking for those kind of solutions that are most plausible or most feasible. There are several other applications, the recommender systems that Netflix uses or Amazon uses to say what you're going to buy next or what you're going to watch next. Image recognition, it's been used for drug discovery. So there are a lot of applications of Boltzmann machines, although most recently it's been overtaken by modern networks, convolutional neural networks, and LLMs, and so on. But Boltzmann machines are getting some resurgence right now because of quantum computing, which I'm going to show later. So here's one of the examples of application to recovering missing data. So if you have an experiment where you have image, in this case, the image was done of a deposition, chemical vapor deposited sample, and then if part of it is missing in your image, you can reconstruct it using a Boltzmann machine. I want to go, I'll probably skip some slides because we're coming to 3 o'clock, so we have a panel next. But I want to show you some contribution of Hinton to backpropagation. Backpropagation is a little bit of a separate concept to Boltzmann machines, although Boltzmann machines also use gradient optimization, but the way of finding those gradients was popularized by Hinton. The idea of gradient optimization is you start from some higher energy, higher cost, and then you go follow the gradient directions to go towards local minima. Those gradients are computed as some derivative of your cost with respect to your weights in the network. So your neural networks has several weights. Those weights tend to be matrices. You try to find the derivative of cost with respect to those matrices. And then you try to move in that direction, move in the steepest descent direction, for example. This is a backpropagation example. A lot of math here, but very simple math, nothing much here. This is all chain rule. So if you've done calculus, you can follow this. The idea is you have some output, you have an input. If you want to find the derivative of the cost function with respect to all these weights, and then you want to go find what's the best weight for this input to give that output, you have to minimize the cost function, you take derivatives. At the end of the day, you get these products of weights. You would always get a product of weight matrices, lots of weight matrices. In a deep neural network, or a MLP, for example, with several layers, you would end up having weight matrices that are trained, and you just get a product of these weight matrices. And this is what Hinton really did, GPUs. GPUs are really good at this, multiplying these matrices out. And Hinton's major contribution really is his AlexNet, which revived deep learning. Using GPU training, using these, he didn't use this, but modern GPUs use large data centers, lots of GPUs. But he obtained a top 5 error rate of 15%, which was the second best outcome was 26%, basically destroying the field, every other approach, boosting and all others. So he has this really took off, and beyond this, we have all the modern advances in neural networks, deep networks, large language models, all of them came from this singularity, which we call AlexNet. And in AlexNet, you use backpropagation. What you do is you take these weights, you have to look for these products of weights all the time, and then when you take the products of weights, you use it for backpropagation, you find the new weights and go on. So that's why GPUs are extremely useful, because you do these multiplications several billion times. My last slide would be just to show my own work. It's a plug, just to show my work. So recently we've been working on Boltzmann machines on quantum computers. The problem with Boltzmann machines, of course, is sampling. It uses contrastive Hebbian, which needs lots of samples to get that partition function, which is extremely difficult to get. So Hinton actually popularized restricted Boltzmann machines that has no connection between the visible nodes, which means you can sample each node independently. You can run this algorithm in parallel. You can use GPUs, run it in parallel and do it. But what happens with all connected Boltzmann machines? In these cases, it's extremely hard to learn. And that's where quantum computing is useful. So there is some resurgence in Boltzmann machines because quantum annealers, for example, can directly sample the Ising model. So you can actually think of an annealer as sampling a quantum Ising model, or in the worst case, just the Ising model at really low temperatures. And you can generate lots of these samples to get the partition function. That's really cool that you could do this, and then you can generate gradients for Boltzmann machines on quantum computers. So there's a bit of resurgence of Boltzmann machines right now. I want to just conclude by summarizing what Hinton and Hopfield had done. Very significant contributions to computer science, but they were motivated by physics. An interesting thing is there was a lot of controversy on this award because this did not go to either particle physicists or astronomers, but it went to artificial intelligence, which is, in a sense, the zeitgeist, what is currently there, popular, but it also aligns with the chemistry award, which was also on an AlphaFold, which is neural network approach. Neural networks actually draw a lot from statistical physics. In a sense, this award was physics for AI, because physical concepts, Ising models, Boltzmann machines, all of them basically played a role in the advances towards Hopfield networks and Boltzmann machines, although they're not very popular today, but they might get a resurgence in the future. But those were the foundations of neural networks. So these inventions didn't stay in labs. They're also transforming our world, and that's really the key for these kind of Nobel Prize awards. So I'll stop here. I think you can ask questions when I'm in the panel, so we're running out of time. So just stop. Yeah.
Yeah, thank you very much, Veera. All right, so what we're going to do now is we have refreshments outside. So, you can take the opportunity to talk to the speakers in person and also enjoy the refreshments. And then in about 10 minutes, we're going to be back. We have some prepared questions for the panelists, and then we also have an open discussion session. All right, we have a very sharp stop at 5 p.m., I'm told. So I think we should get started with the panel, and people will come in. So we have, again, 4 very distinguished panelists who are deep thinkers at the intersection of AI, science, computation. And as I said, I have some prepared questions, but we also have an open session that we will have. All right. So I want to start in maybe this order. I mean, it would have been better if we had been mixed better, but that's okay. All right. So the first question, again, is for everybody. So each of you talk to us about some of the most interesting questions in your field that is driving science. Okay. So Charlie, we start with you. Yeah, it is
now
on.
It's on.
Yeah.
Now it's on. Okay. Machine learning is really pervasive in biology and chemistry, and there are many, many areas that are being impacted, from sort of drug discovery to property prediction for both chemical things and biological entities. Yeah. It's touching everything at this point. In fact, it was interesting. I was sitting down at lunchtime today looking at my phone, and I got an email from the American Chemical Society, and there's a big thing coming out in CNE News about machine learning and its impact on chemistry, and there's both angst and, I think, excitement about what it's going to do. But it's touching everything. It's hard to, you name a topic, and I can probably tell you what it's doing there.
So your answer to my question is machine learning is really driving science in your field.
Well, it's certainly being utilized in the science. How it's driving, probably it's driving science, but it's still, I think many people are still in the area of questioning and wanting to understand what its limits are. In our own laboratory, for example, we're looking at, in the context of drug discovery, some of the docking methods, the machine learning-based docking and scoring methods, and whether they're really good or whether they're just able to work within the scope of the things they've been trained on.
Okay, all right, thank you. Same question to you, Indika. If you want, I can show your one slide.
So yeah, maybe Karthik, I'm
Sure.
having a hard time here sitting. So yeah, thank you. So I just put a slide to show you. So I'm so excited now this field. I'm a little bit new to this field. I'm coming from engineering, math to biology. And last few months, no, few years now, I'm so much into AI. But this is a quote from Demis Hassabis. I really like this one. This is what he said was here, understand what, if you only understand what you are going in biology, if you understand the dynamics and the interactions between the different things in a cell. There are two parts, dynamics and networks, right? So that's so exciting to me. That is something true to my heart, is to study dynamical systems and network systems. And the last thing I hear in this slide I want to share with you, this is one of my favorite papers that when I switch from to biology. If you see the first author is Leland Hartwell. He got the Nobel Prize in 2001 in discovering cell cycle checkpoint. The second author is John Hopfield. All right? I highly recommend you to read this paper. Highly. This gives you so much idea still, you to think, right? And my next slide is little bit to tell you, to answer to Karthik question. So where the field is? Oh, sorry. Yeah. So keep this close to you. Yes. So and this is an active project I have funded by DARPA. We call Twin Cell Blueprint. That how to bring AI-assisted cell reprogramming. So what is cell reprogramming? Cell reprogramming means how to, let's say, a skin cell in your body. Can I change the skin cell to something else? Because every cell has the same genome, right? This is a project I am so much involved with last few years. But still, there are a lot of open questions in this area. So I want to use and I want to excite young people to bring the aspect of AI to this problem, cell reprogramming. But that required also mathematics, like dx over dt equal fx, and plus say bu if it's a control. But still we don't understand genome dynamics, right? That is maybe we can, with modern AI techniques, maybe from data we can approximate that function. The other one is, as Demis and as Charles mentioned, protein, how protein will dock to DNA, right? So there are a subset of proteins are called transcription factors. Those are the ones come inside to the nucleus and do something, right? Still a big mystery. That is a very dynamical system. It's a dynamical system, right? So that's something I'm actively working, and I'm so excited to use this modern technology. But my last thing is, Karthik, everything we do, I think we need to humanize our science. There are a lot of problems we still need solutions. Like, can you reprogram a cancer cell, right? So anyway, that's Karthik, I want to introduce this is my area.
Thank you very much. All right, so Veera, what's driving some of the questions in your field?
I work in computational. I work in computational material science, but I also work with a lot of experimentalists measuring materials. So there has been a significant amount of work going on in neural networks for materials design. One of the most popular ones, MatterGen, that Microsoft recently did, where they talk about crystal structures. You can model them as graphs. And then you have DFT data. From the DFT data, you can try to predict new materials or try to predict new phases. So that's getting popular. If you ask me, it's been successful. I would say it'll be successful when they design a new superconductor. It's not happened yet. I think the drawback is we don't have the theory yet, the theoretical accuracy. For example, when I talked to Professor Gavini in the mechanical engineering, he runs this DFT simulations getting up to 0.1 milli-electron volt in accuracy. With AI, you don't reach that accuracy. You can train a lot of AI, millions of DFT calculations, it would be very hard to reach that accuracy. So it's been quite a challenge designing new materials, but it's an ongoing research area. Also, large language models are being used for material science. I have a colleague, Venkat Vishwanathan, who's doing a lot of work on smile strings, designing polymers using, think of polymers as smile strings, which are, you can think of carbon attached to hydrogen as a string, and then you use large language models to try to learn chemical structures out of it. That's the second. The challenge there is, again, when you have large polymers, you have very complex structures. There's no representation for cross-link structures, for example. So that's a challenge. The area it's been most successful in my domain, in materials domain, is imaging. In imaging, it's been wildly successful, artificial intelligence. So now you can actually image atoms, do a TEM on a surface, and then start predicting what's happening. If there is missing data, you can fill the missing data with AI. Most likely, it's greatly successful in those areas. It's useful for 3D reconstructions. Tomography, for example, if you measure 5 slices, can you reconstruct the whole image? And it's been very successful in that. I've been translating that to biology also. In imaging, it's been quite successful. Recent area of research has been robotics. So there's an interest in robotic labs where chemistry is being synthesized by robots. So there is no graduate student. So the graduate student switches on a machine and goes. It works all night, mixing powders, mixing alloys, heating it up, trying to see if there is new materials being generated. So you see these computational arrays that robots do. That's getting popular. Not much has come out of it yet, although some of my colleagues are designing electrolytes, for example, but that's a very cool area. The big drawback of all of these techniques for designing materials would be lack of data sets, lack of reliable data sets, and the second issue is non-explainability of AI, because you design something, you cannot really explain how AI is telling you the answer. There is no, the machine learning algorithms are not explainable. So that's also a big challenge. So that's all I had. Yeah.
Right, James.
Yes, so I'm in elementary particle theory in the physics department. So I'm coming from a rather different perspective than some of the other panelists. And I'm going to simplify a bit, 5 areas which are really high inquiry areas in my field. And here's one, the first one. If you put your hand out, you ask yourself how many atoms are in your hand, and it's something like, I don't know, 10 to the 30 or something, Avogadro's number times volume. Within each hand size, there's approximately 1 dark matter particle on the prevailing idea of dark matter. Just 1 out of 10 to the 30 or something. That 1 dark matter particle is absolutely critical for how our galaxy is rotating, how the expansion of the universe is operating. There's firm evidence that dark matter of some kind exists. We don't know what it is. There's a huge mystery. It is one of the dominant questions in physics departments. The second is, imagine you're at the early universe and there's a big explosion, and you know how things develop in a big explosion. You make a particle and an antiparticle. That's what big explosions do, fundamental energy before you have matter. You make both in equal numbers. Well, in this room, you're all matter. There's no antimatter. If this table were antimatter and I hit my hand on it, it would be the most powerful explosion in the history of this galaxy, okay? There's no antimatter out there that we know of. Why did that happen? Huge mystery. We don't understand. We have many ideas. There's experimental inquiries for it, but we don't know. Another, at any given moment, I will be Carl Sagan for a moment. There are billions and billions of neutrinos passing through you at every second. Those neutrinos are very elusive particles, very hard to study. You need detectors the size of Michigan Stadium filled with water to see Cherenkov radiation, to see 1 interaction per macroscopic time unit. Okay, that's how weakly interacting it is. It was discovered not long ago that they have mass. And if we had a panel here 50 years ago of particle physicists and other physicists, they would have sworn by their entire paychecks and their life savings that neutrinos were massless. How did they get mass? Don't know. We don't know how they got mass. We're still working on it. So one of the premier projects in particle physics in the United States is to figure out that, help figure out that question, okay? Two others very quickly. One is quantum mechanics at very high energies attached to gravity. How do they meld together? We don't fully know. We have really good candidate ideas and these string theory approaches. They lead to, if you follow the path for a few decades, they lead to problems that are similar to protein folding problems. 10 to the 500 minima. Where are we at in this minima? How do we decide how we got into our minima? How our present universe looks like? We don't know. We're investigating that problem. Last one. Higgs boson was discovered only 10 years ago. 10 years ago, in one day, there were articles on the web saying that the Higgs boson does not exist. So experiment was absolutely critical to establish that the Higgs boson was there. The Higgs boson gives mass to all elementary particles. Well, where did that mass come from? Why did the Higgs boson exist? And in particular, why is its mass so extraordinarily light compared to a characteristic scale of black hole creation? If you follow through basic quantum field theory arguments, which is the most basic fundamental description of nature is quantum field theory, it tells you it shouldn't be that light. It gives you really strong reasons to think it shouldn't be light. Why is it light? Huge issue. We're trying to figure that out.
Wonderful. That's 5 homework questions for you. Due on Monday. All right. That's really good. But now we'll go this way, and I'll ask you the hard question. How is AI, or how can AI help answer the questions potentially?
So in my area, in my expertise, I really love collider physics on answering many of those questions. Many of those questions that I described have a high energy collider experimental approach for determining the answer, trying to determine the answer. And high energy experimentalists, both in the US, at CERN, other places in China, have been on the forefront of utilizing machine learning and AI techniques to take extraordinarily complex data. The data is, if you could write it all to tape, at CERN alone, it's 1 petabyte per second. The detectors are bigger than the size of this auditorium. You cannot write all of that to tape, so you need extraordinarily fast electronics. The extraordinarily fast electronics to decide what to put on tape is fed through a machine learning system to, it's basically a classification problem. What is worthwhile versus not? So that's step 1 of AI. Step 2 is you form a cajillion little flickers in your detector all throughout a volume the size of this auditorium. In the old days, what you had to do is say, I think those cluster of flickers are an electron, and I think those cluster of flickers are a quark, and then you get some rough estimate of what's going on. Today, you can train these complex detectors on simulated data and on real data through bootstrapping mechanisms to not even worry about the ontology of the structures, but just what are all the things that flashed and how does that connect to the theory? And that has been the most recent revolution in how AI is increasing accuracy and increasing the power of discovery in these different areas that I talked about earlier.
Yeah, thank you. That's very concrete. Yeah, I was gonna skip you both because you did show something about how AI is impacting your field, but because you picked up the mic, if you can maybe do a quick. Yeah.
My answer is very short. So unlike CERN, the issue with materials is lack of data. CERN has an enormous amount of data, particle physics, and materials, the big issue is lack of data.
Yeah. So Indikat,
man.
talk about, you spoke about the problems you're interested in, right?
Yeah.
Talk.
So
Oh, he's
I will
on the
draw
board.
a picture here.
Now talk about how AI can help.
Yeah. So let's say this is the
Keep the mic close to your.
this is
Yeah.
your genome. Right? Your genome is inside the nucleus, this tiny volume. So the proteins, some proteins come inside. They come as a activate something and suppress something. These are the proteins. It's still unknown how this the activator or a repressor, how they dock. This whole thing is changing with time, right? I think AI will help us to understand both how the docking problem, protein, dock to DNA, and not just the one protein. It's a family of proteins come together and change the state. I think this is the way I am excited. I think the future will be to understand this one. AI will help us.
Because the audience is broad, if you're able to understand that, how does it help humanity, for instance?
Yes, I will tell you one example. So if you have say a leukemia, it's still a treatment is a high dose of chemotherapy. If you get a high dose of chemotherapy, you wipe out your immune system. To rebuild the immune system, you need to do a bone marrow transplant. Bone marrow transplant is the treatment for the treatment. Let's say I got a leukemia, got a high dose of chemotherapy, and then I need to rebuild my immune system. To do that, I need to find a donor. Let's say Veera is my donor. He will give me a certain cell type called hematopoietic stem cells to rebuild my immune system. There's a problem with that one. After 3-4 months after that, if you get his cell put into my body, his cells start to attack my body. So that is called graft versus host disease, GVHD. Imagine someday I don't need Veera's cell. I use my own cell and reprogram and put it back. That completely humanized our science. That's a big problem in bone marrow transplant. Even to find a donor is hard.
So reprogram your own cells.
Own cells
Yeah.
for solve problems.
Okay, thanks. So my question to Charlie is somewhat related to your talk. I mean, because you clearly answered the question of how AI helps your field, but I have a relevant question there. So, in computer science, there is this thing called, Sutton's, what do you call, the bitter lesson, right? It is, the bitter lesson goes like this. Initially, we use all of our physical intuition to build models, and we make some progress, and then AI comes in, and it basically takes over and makes us all look silly. Okay, I'm just being tongue-in-cheek here, but that general argument is used, right? But in the case of AlphaFold, for instance, you only had 170,000 protein snapshots. And from the 170,000, they built this model, right? But instead of having trillions of those, right? But the reason why it succeeded is because they, as you mentioned, they used a lot of physical principles. If they did not use the physical principles, they would need 1 trillion data snapshots. And getting one data snapshot is like a PhD's worth of work, right? So anyway, long-winded way of asking the question. I mean, AlphaFold, it worked beautifully, in protein folding. What are other problems where, like some of these, like what Veera said, we are data scarce, and if you just rely on AI, you cannot solve the problem. But then by bringing in physical intuition, inductive biases, we get a huge leap in capability without enormous amount of data, right? So for instance, ChatGPT and all these foundation models, they succeeded only because of enormous amount of data. But AlphaFold came through without that. So what are some other problems in your domain where with more or less existing amounts of data, big transformations are possible?
Well, I'm probably going to start out by disagreeing with you a little bit about a point.
I'm very glad to be
about
corrected.
the 170,000 pieces of data, the protein structures that are known. And make an analogy with basis functions and basis sets. If, in fact, I have a complete set of functions, then I ought to be able to express anything with that set of functions.
Sure.
So some would argue that, in fact, within that 170,000 structures, the entire world of protein fold topologies are there.
Okay.
And so they had everything they needed to learn.
Okay.
Okay. However, to get to your question, I mean, one of the areas that I'm also currently quite interested in working in is in biocatalyst design. And I think machine learning can strongly impact this question of sequence to function there. But the problem is that the data for learning, where suppose that you want to find a biocatalyst that carries out a particular chemical reaction, and you want to use machine learning to look at families of sequences to suggest which ones are likely to carry out those chemical reactions, you've got to have the data to train it against. Well, the sequence data may well be there, but the chemical outcome data to do the training is not there. And this is an area where I think we can think more about it. And one of the projects that we did, we tried to combine physical modeling with the machine learning to help predict outcomes. Okay. But also here, I think, is a place where more technology development will aid and bring us to making AI helpful for those sorts of problems too.
Okay, great, thank you. So I have a few more questions, but I want to open it up to the audience. Whoever wants to ask a question can come up. You have a question? You're passing the mic. Anybody has? We have physics, chemistry, biology, medicine, computational science, materials. We have all that expertise sitting there. So anything, any question is fair game.
Yeah, thanks for the presentations today and really thinking it through. So one of the things that I find, after now having used AI a little bit, fascinating is that artificial intelligence is not really intelligent. And you touched upon this a little bit because you emphasized how it's interpolating. It's very good at data analysis and sifting through data, sorting things that it has seen before, that it has been trained on. Sure, they are foundation models. They can be trained with synthetic data, and they become better and better. But in the end, I feel so far, it's still not the matrix. It's still not quite what people imagine it could do in the future, being independent intelligence, right? That we need a Tom Cruise to fight, right? And so, so the question is, can that be more emphasized? Because, of course, in a broader society, and also among some of the leaders of the field, there has been this monster built up that it would be a very dangerous future, when in fact in science, certainly, it helps a lot with those kind of things that are difficult to do for a human and can be really powerfully supported by AI. So where's the balance? What do you think about the intelligence of AI and so forth?
About 15% of my work is in defense-related areas, and there's a huge amount of discussion on this. Actually, they're worried that Tom Cruise might need to be involved. So the big worry is you don't have to be terribly intelligent to be terribly damaging. And we all know this pretty well. And in a combative kinetic warfare situation, the worry is no matter how high of an ethical level we might have for it, the adversary may have lethal autonomous weaponry, all the way from robotics to whatever. It turns out that intelligence, in a general's mind, this is what I've learned at some of these security conferences, intelligence equals certainty. So if your autonomous object, you are certain that it will behave the way you want it to behave, it is intelligent. That is functionally how they would view it. If there's uncertainty, that's uncertainty associated with algorithms. Not, we have uncertainty associated with emotions, and which actually in these conferences they talk about human emotional uncertainty is perhaps more problematic than the uncertainty of some deep neural network trying to decide whether or not to shoot somebody. Those are just a few of the issues, and there's many more. I could go on for a long time.
I think you can, Karthik might be better off answering this question, but just give an example as this collaborative combat aircraft for DoD. It's going to be it's called F-47. There is an aircraft that works with 2 drones. The drones are going to be controlled, although they're going to be autonomous. They're going to fly ahead of that aircraft. A few miles ahead of the aircraft, they're seeing what's out there, but they will be controlled by the pilot in the main aircraft. So they're not going to let drones decide whom to kill. There would still be a person sitting at the back who's controlling it. Yeah, we are not at there. Karthik, go ahead.
But I don't think we still got to the core of this question, right? How about the intelligence, real intelligence in these algorithms? I would like, if you want to take, but that's a different, we can talk later. Maybe second question.
Could you say something about the inverse problem for protein folding? So given a desired structure, is there a way to, what's the state of the art for finding the sequence that produces that structure?
So that's called the inverse folding problem. It certainly continues to be an active area. I will also say that early on in the CASP exercises, that was largely the approach. They took the structures that they knew, and they did what they called threading and tried to find out whether this sequence was compatible with that structure. So that continues to be an ongoing research area. And I think we've gotten much better at it because mainly we're able to build very large multiple sequence alignments, and we're able to learn much more about what sequences are similar so that the matching then of a sequence to a structure is gotten to be easier. And I think machine learning has played some role in that, yes.
Can I make a comment? So I'm so fascinated by, in my body, this is happening every second. Right? The protein fold and do something. So I think that's nature somehow have an amazing algorithm, right? And do we still know that algorithm? That how nature do that? Every second, that's many proteins fold, and that's me, right? I think.
That's following instructions, right?
Yeah, some algorithm has to be some algorithm. I don't know whether we still know that. Right.
Other questions? You want to come forward and ask? Maybe you're closer. Maybe you can ask first, and then we can.
Thank you. So I would like to know that is there any tricks or paradigm that embedding the knowledge in scientific computing into the AI model?
I'll repeat what I think the question was, which is there any scientific principles embedded into the AI systems? Yeah. Yes. So in the case of the example I had about CERN and the data coming through, there's all these things lit up, and different particles will have different speeds depending on if they're down the decay chain. So what they have built in, they've had some sort of physics-informed neural network type approach where what's called Lorentz invariance is put in there. So they will take the data, train the network to recognize if something is moving at a different speed, and they will boost it back to at rest or at some pre-described notion. And that is based on an absolute sacrosanct principle of physics, which is Lorentz invariance. They would not dare to do it on a principle that is not sacrosanct. Because there's a lot of other things they could have manipulated the data on that we think are correct, but they're soft theorems. They're not really rigorous, super rigorous. So, so that's one example. Yeah.
For proteins, some of the amino acids after translation gets modified, and how does those affect the structure folds predicted by the alpha fold, and also depending on the time point within the cell, those modifications change with time, and how does those can be predicted?
Yeah. So, yeah, that's a very, very good question. I mean, so the point is that actually protein sequences throughout their lifetime, if you will, can get modified shortly after they're made. For example, they can have sugars and other post-translational modifications added to them. Largely, I think that the working hypothesis is that the dominant organization of the structures aren't strongly affected by that, but how instead they interact with other proteins is what's really being affected. Those are modulators of protein-protein interactions. So in the context of protein structure prediction, as an individual structure prediction, I think largely that's not coming strongly into play. In the modeling of protein-protein interactions, which of course is the forefront, I think, of where AlphaFold 3 is and some of the other machine learning methods directed towards predicting the complexes of proteins. That's a critical factor, and we don't really understand because largely we don't see those post-translational modifications often in the final crystal structures. They're not in the amino acid sequences. And as you said, they change during the course of the lifetime of a protein within the cell. So for the most part, we're largely ignoring them. Where they're going to be very important, and maybe the current frontier, I think, is in protein-protein interactions or protein nucleic acid interactions, just to make my friend happy here. And I think that's a current frontier.
So you've talked a lot about how neural networks have been very successful in making advances in science and all that. So do you think neural networks alone are sufficient for making further progress, or do you need to bring in what's called classical AI, which is symbolic manipulation and that stuff, and combining these two is sometimes called neurosymbolic AI? Do you think that would help make more progress, or do you think neural networks alone are sufficient for making this kind of progress?
Yeah, that needs to be. Yeah, so there needs to be multiple modalities. So we can't just stick with LLMs. There needs to be research in multiple areas. There's been recent resurgence in analog networks where you just don't have weights, but you have continuous functions that are fitted, or Fourier transform-based neural networks. So there needs to be research in other areas, mainly because AI takes a lot of computational power, and any simplification will really improve AI.
So my question is more of like looking for this dark matter particle. So how can you like sift through all of this data and find that one little outlier that you can say for sure is either like a dark matter particle or whatever you have of interest in comparison to the large amount of data that you have?
Right. So in the case of CERN, they're looking for dark matter being made in a collision. You go from petabytes down to about 100, 200 gigabytes per second, which is enormous, still enormous. You might have out of 10 to the 12 events that you can record, 10 to the 11, 10 that would be dark matter. And the way you do it, a human can't do it. There's just a paper published, your question is a paper was just published 5 days ago on how do you do this with AI. And what you do is you train and train and train the hell out of it on the signal with the background, and then you just throw it and said, and this is really not something particle physicists like, but throw up our hands, figure it out. Dear deep neural network, figure it out. And then an answer will pop up. We found it or we didn't. And then if you found it, then you go back to the old classic ways. You look at the events that it says were the correct events. You look to see if, oh, did somebody spill a Pepsi on the control panel at that moment? Or you do all these additional checks. But that's where we're at now. You just throw a cajillion events with a cajillion amount of training and say, please tell us.
Yeah, sorry. Just a follow-up question on that. So when we have such a large amount of data and we are trying to find pattern in it, are we trying to mimic the human expert who's just annotate like what the pattern we are looking for, or we are finding some unknown pattern that can be otherwise learned by machine? Also, the second part is, for such a large amount of data, I would assume if there's a pattern, it would converge quickly, or the challenge is we need to have like many different experiments to say we actually have the pattern.
Right. It's a great question. So physicists, the way CERN was originally, and particle, I keep on saying CERN because I was staff there for 6 years, but the way they approached that was, let's make ourselves superhumans. And boosted decision trees are basically line up millions of people and have them say yes, no to questions when data comes in. And you already process the data a little bit. What is a quark? What is a jet? What is a photon? And then you ask yes, no questions. You make this boosted decision tree. So they did that for years and years. But the advancements of AI and deep neural networks, they are abandoning that to some degree. And they are just saying it's too much. It's too complicated for us to, in fact, it does better when we're not thinking. It does better. Only a little bit of thinking, like this Lorentz invariance. It does better if we allow it to do its magic in this deep neural network, and it will effectively come up with many different categories of how you cut and slice data that we humans just can't think of. And only after the fact do they root through the answer and say, I can see some principles. That's a clever move. It's like in that, when the chess, AlphaGo.
Yeah, I was just going to say that.
It's just like AlphaGo. So at the end, it made a move that no human ever would have made, right? And this is exactly what these deep neural networks are doing on this discovery process. It's making moves that a human wouldn't do. And answer your second part very briefly, there are 2 experiments. And they're running cross against each other with different algorithms and different approaches. And we really don't consider things a discovery until both of them converge. Yeah.
Thank you.
Thank you. Wonderful panel. So my question is more related to the material scientists as well as the protein engineers over here. My question is regarding AI and its application towards what you've actually shown in the presentations earlier, which was more of a thermodynamic constraint to how structures were being generated, or you're basically testing against. What if there were kinetic components to the structures which have never been included in the AI training set? How do you expect AI to actually start learning all of these as well? Because kinetically trapped structures are also possible in real life, and these do not normally follow the thermodynamic principle of finding the minima as well as finding where the local gradients are. So there are certain aspects to it which you cannot train the AI to not normally. So how do you account for these sort of noises in the structure?
Good question, because there are some examples I can give. For example, if you want to predict a crystal shape, then you could use minimization of energy, but then that solution never really matches what you see in an experiment. That's because a crystal shape takes a million years to minimize. So think of diamond. Diamond is metastable. You can predict it's a stable structure, but it's still metastable. It's going to eventually become carbon after a million years, billions of years. So that kind of kinetics is missing in current models. To include kinetics, we need to have better models, better physical models of kinetics. We need to understand what are the growth velocities of facets, for example, in crystals. Some facets in a crystal grow faster than others, but eventually they will minimize the energy. They get the right shape, but then in a metastable state, you can get different shapes of crystals. So that's a very challenging problem, but it needs quality data to do, and it also needs good simulations to generate those kind of data. And to generate good simulations, that's where AI really comes in, because you could use AI to improve upon simulations. For example, if you're doing DFT and you're predicting some answer, that DFT answer can be improved if you can do a lower scale simulation, like quantum chemistry simulations. You can extract exchange correlations from quantum chemistry, put that in DFT. Through machine learning, that can improve your simulations. And if the simulation data can be improved, then machine learning will improve for material science. Yeah.
Thank you very much. So we have to stop here because there's another group trying to come in. So if the panelists have a few minutes outside, maybe there may be a couple of additional questions that people wanted to ask. But outside of that, thank you so much for a really engaging. Thank you.